Each year, the Society for Developmental Biology presents Lifetime Achievement Awards to honor a senior scientist who has made exceptional and lasting contribution to our field. This award reflects our long-standing partnership with Developmental Biology Journal, recognize not only groundbreaking research or educational impact, but also a deep commitment to membership, advocacy, and the service to broader community. And this year's recipient is Dr. Professor Billie Swalla at the University of Washington. Okay, Dr. Swalla began her journey at the University of Iowa, then moved on to become an assistant professor at the Vanderbilt University, and she later held a position at the Penn State before joining the University of Washington in 1999. Since then, she continued her research both at UW and at Friday Harbor Laboratories, where she also made history as the first female director. Okay, so Billie's scientific career as a developmental biologist started early at MBL, and this is where she began her lifelong pursuit of one of the big questions in biology, the origin of caudate. She used ascidians and hemicaudate as her model system to tackle this fundamentally important problem. And this photo shows Billie right in the middle with that big, unmistakable smile. And at the time, she had just finished her master's degree studying cartilage and muscle development at the University of Iowa. She was 25 years old, single parent, and came to the MBL course with her 3-year-old son, Tony. Billie said that this was the very first time she saw the ocean, and as you know, Billie, of course, swam in it. And also she saw first-time ascidian embryos, and it was love at the first sight. Billie's research has focused on the evolution of caudate body plan, especially through her work on tunicates and hemicaudates. Studying her development provided insight into the ancient caudate body plan and the evolutionary steps that lead to vertebral innovations like notochord, neurotube, and tail, particularly by comparing the notochord and tail formation in tailed and tailless molecular ascidians. She has also contributed to understanding the phylogenetic relationship among tunicates and hemicaudate, and she helped to reconstruct the important evolutionary tree that connects them to vertebrates. And Billie is always on the go and busy doing sciences. If she's not in the lab, she reads papers, encourages science to everyone, and she's on the field doing the mucky hands-on work. And she's excited to be out there, but her students might think otherwise. Most people, one might say, most people go to the beach for sun and surf. And the other might say, I feel like I've lost my motivation. Or others say, do I smell like low tide? Always. And Billie is so optimistic, but she thinks otherwise. Wait, don't step on the puddle. There are at least 4 species of tunicate in it. Or she might think still better than the windless lab. At least out here, ascidians still appreciate me. And this is the last slide. This is the photo of Billie at one of the tunicate meetings. She's raising her hand as if she were championing tunicate and hemicaudate. And she is perhaps saying, Tunicate may look like a sad little sea squirts with identity issue, but they haven't given me the endless joy, surprising insight, and just enough data to keep my grants alive. But, she told me that this is not for that. She was actually imitating John Travolta on the dance floor. So please join me in welcoming an incredibly dedicated developmental biologist, an inspiring teacher, and a true community builder, a lifelong champion of Assyrian biology, and yes, an absolutely unstoppable dancer on the conference floor, Billie Swalla.
Thank you so much. I'm kind of overwhelmed by that wonderful thank you introduction, Ken. It's just been such a fantastic meeting. I think we should all thank the organizers. We all know how much hard work this is to put together a huge meeting like this. And thank you, thank you, thank you. So. Absolutely fabulous. Okay. So let's see if I can find the pointer here. Can you see that? Yeah. Oh, goody. Okay, so I'm going to talk about deuterostomal ancestors and chordate origins. And Ken already went over this, but I've worked on lots of different species during my time in science. And now I've really narrowed it down to a microevolutionary scale. So these are two species that are closely related. We can actually see their go into the genome. We can align the genes. We can see where the promoters have changed on them. And I think of this like a scale. If you're on a lawn and you're seeing the blades of grass, and you can compare the blades of grass and figure out what's really changed here. And then this is what I really am excited about, is this macroevolutionary scale. And that's like if you're in a plane looking at the lawn and you can't see the blades of grass. You're too far away from them. They're long gone in evolutionary time. But you can still see patterns, and you can still try to figure out what's happening down there and how did that work. And so my lab has really been focused on that for the last however many years. So I always put a little thing in here for Friday Harbor Labs. So I'm now full-time at Friday Harbor Labs. It's on San Juan Island, so I came from San Juan to San Juan, and I'll be going back to San Juan. That's quite a long trip, let me tell you. But like all marine labs, it was put there because of amazing biological diversity, and there's courses there if you're interested in students coming there and seeing it. And anybody who comes at any time, I'll give you a little tour of the island and good wine and et cetera. So you're all welcome there. And there's also facilities to teach classes there. So I teach many classes there as well. So I want to first thank my graduate advisor, Mike Solersh. Some of you may remember him. Unfortunately, he died of AIDS at 53 years old. And he would always say, do the right experiment. Think about developmental mechanisms, not just your embryo. We had both sea urchins and chicks. I did my PhD working on chicks, but we had sea urchins in the lab. And he would never let us say in the chick or in the urchin. We had to say what we were studying, what mechanism we were studying. And I think that really influenced my research. Keep your microscopes covered. I still tell my students that all the time. Seawater here, keep the microscope covered, put that cover back on there. Eat chocolate every day. Michael loved chocolate. We always had chocolate in the wild. Yes, absolutely. The other person who loved chocolate was Rudy Raff, and always at a meeting like this, I'd hear a crinkle, crinkle, and Rudy would have a big chocolate bar, and he'd give me a little piece of it sometimes. Don't lose your career when you have a child. Michael had a child the year after I had my first son, and so he was a lot nicer to me the second year when he had his own kid than he was the first year. And this is just to tell you that you don't always have to take everything your mentors say. Michael was Jewish, so he thought everyone should work on Christmas. And I used to tell him, I know, Michael, but it's a holiday for a lot of people, okay? So I'm going to take off Christmas Eve and Christmas. And he'd be like, oh, okay. So I really think of my career as starting in 1983 when I went to MBL to the embryology course. And for those of you who are excited about marine embryos or any kind of embryos, the embryology course or any kind of course you can take is really a fantastic opportunity to do that. As Ken told you, I went with my young son, and that's also the first year I published my first paper. 2 distinct classes of prechondrogenic mesenchyme and embryonic limb buds in developmental biology. Woohoo! And I've published many papers in developmental biology since then. And why did I go to Woods Hole? My family thought I was nuts, by the way. I grew up in Iowa, and nobody had been to the East Coast. My dad told me everybody gets murdered there. And I said, good, I'm going to go get murdered. And he said it's wall-to-wall city. And so when I got to Woods Hole, I was like, this is not wall-to-wall city. This is beautiful. And I went there for Ascidian tadpoles. And the reason is the Ascidian tadpoles have a body plan, just like you all do. You're all chordates. So they have a head here and a tail with this beautiful 40 notochord cells here. And they don't have pharyngeal slits at this stage. They're non-feeding. They have pharyngeal slits once they metamorphose. And they have this beautiful nerve, hollow dorsal nerve. So someone said to me, hey, you showed this last time. And I was like, oh, man, I'm going to show the same thing. But it's a great movie. This movie was done with George Van Dassel when we were teaching an embryology course. And this is what I went to Woods Hole for, to see these beautiful Ascidian embryos. This is Corella, which is clear, and so you can't see the colored cytoplasms that you can see in other species. You can see the plane of bilateral symmetry, which is right down here. So Ascidians have bilateral symmetry. It's a very unusual kind of cleavage that's found only in Ascidians. Oh, please don't do that to me. Come on. Let's try this again. I'll try not to touch it now. Okay, I'll just tell you about it. So don't look at the outside. The embryo's on the inside, and then you can see these test cells moving around. Don't look at the test cells. They're moving all around. Those are somatic cells that crawl in after the egg is made. And then outside of that, you see the follicle cells. So the follicle cells are out there in every Ascidian embryo as well. They're the outside cells there. You can see that bulletproof chorion, which the only thing that is not good about Ascidian embryos is that you have to get that dang chorion off to do anything for them and to do in situ hybridizations. But we know how to do that now. And you can see now, again, this plane of bilateral symmetry. I'm going to try to do this without moving it ahead. Those are the big endodermal cells. We're looking from the animal pole, so what's happening is it's gastrulating. The endoderm move from the vegetal pole, and they move up into the animal pole. And then the next thing that'll move in is the pre-muscle and then the pre-notochord. All Ascidian embryos have a very similar lineage, so I can look at one in the plankton and tell you where the different cells are. This one here, oh no, don't do that. Oh, it keeps starting over. All right, we're going to have to move along here. This one's starting to, it'll start to neurulate. See if I can get it to go here. And so when they neurulate, they'll roll because of the cilia that they have, and you'll see the neural tube. And what happens in Ascidians is the neural tube actually goes from the posterior to the anterior. It's the only chordate that the nervous system rolls up that way. It's going to start rolling here soon, and that's, again, from cilia that are on the outside of it that cause it to roll. It needs that for left-right asymmetry. It has nodal expressed on the left side. You're very surprised to see that because they have bilateral symmetry. Here it goes, here it goes. So the head is to the left. There you can see it right there. That's the neural tube. It's zippering from the posterior to the anterior, which is the way that they close. And now you're starting to see the head's formed over on the left side, the tail's forming on the right side. They have 40 notochord cells. All species we've looked at have 40 notochord cells. They converge and extend into a single stack of cells that's in the tail there. And you can see the tail now is starting to elongate. What's happening is those 40 notochord cells swell, And so they're pushing out the tail. This would be like mid-tail bud now, so you can see the head really well. You can see the tail here starting to curl around. Pretty soon you'll see that we are focusing it. There you can see the 40 notochord cells right through the center of the tail. So it has 40 notochord cells in the center, has 3 rows of muscle on either side. 3 rows of muscle don't correspond with the notochord because they're stretched out. So they're like this when you got the notochord there in the center. And you can see now, got the head and the tail. Don't look at the test cells. They're trying to get your attention. Don't let them do it. And you can see the head now, and soon it will start twitching. So the muscle, it's some of the fastest muscle that's developed in the animal kingdom. Some of these hatch at 12 hours. So they're starting to make muscle, and they're starting to twitch because it's going to go swimming off. And you can see now that it's getting kind of active. And the test cells getting kind of excited now. You can see the follicle cells are looking kind of bad there. It actually has a hatching enzyme that it secretes to get itself out of that chorion. And when it hatches, what it's going to do is they actually don't swim like fish a long ways. They swim up to get to the water current, and the water current is what takes them out. And then when they're ready to metamorphose, what they'll do is they'll swim down towards gravity. So they swim up towards light, down towards gravity. And the other cool thing that when I show this to students, I say, notice that this organism is the same size as the single cell, the fertilized egg. So it turns out in most marine animals, this is the way it is. You can tell that first cell, that egg, was huge because now there's an entire ascidian larva. And there it goes, bye-bye. And those cells are like, where'd they go there? So with the Mogula, what we do is we look at these 2 different species. So unfortunately, the species are also spelled very similar, so it's quite a pain to work with them, but they've got a tailed and tailless species. And what happens is that when you have this egg, you always get a tailed tadpole larva. Even if you fertilize with a tailless sperm, when you have this egg, the tailless egg, you get these little hybrids where they have now an eye spot. And you can see now they have a 20 notochord cell instead of a 40 notochord cell. And we're really interested in what's going on here. What we found is that most of these, many of the larval genes in these tailless species are pseudogenes. So they start to have all these hits. Their muscle actins are pseudogenes. This tyrosinase that makes this black pigment is also a pseudogene. And so what we do know is that this pigment that we see back in the hybrid is from the tailed species, not from the tailless species. So we can tell the 2 species apart, and we can see what's being expressed. And we don't know yet what causes this tail to converge and extend. This is now my 2 sons, David and Tony. This is in Banyuls. And Henri Lacaze-Duthier, this is actually his grave. I didn't realize it at the time, but I didn't let them play on there anymore once I realized he was buried there. But Lacaze-Duthier founded 3 marine labs in France that are still going, which we're very appreciative of. And he founded this marine lab at Roscoff because of these tailed and tailless species. And he said it in French, of course, because he was French. But he held them up and he said to the National Academy of Science in Paris, we need to study these tailless ascidians. They're going to tell us the chordate origin. I was like, yes, Lacaze-Duthier. I love that. And we're still working on them now. For those of you interested in tunicate genomes, which I am, there's an aniseed. You have to put aniseed and tunicate, otherwise you get all these aniseed recipes. You don't want that, but it's run by Patrick Lemaire in France, and we have quite a few genomes on there now, and you can search for your favorite genes, and we can look for synteny between different species and et cetera. So it's really great. And I'm involved in a collaboration with Anna Di Gregorio and Alberto Stolfi to try and figure out, this is that great notochord network here. Brachyury here is one of the very first transcription factors that comes on in notochord. And Yi-Sun Su gave a great talk here that shows that some of those networks with brachyury are very, very ancient, even back to single-celled animals. And so how did the chordates co-opt that brachyury, very ancient network to make a notochord, this new tissue, is what we're trying to figure out. And we're looking at all these different genes in a variety of species in. So how does the role of development and evolution of animal bodies change? I used to tell students I really wanted to study development and evolution, so I had to start a whole field so that I could do that. And it's kind of a joke, but not really, because I was very interested in not just how those gene networks and how animals develop, which we're all interested in, and plants develop and other things develop, but also how do they evolve? How do those networks evolve? How do you get different tissues? And to do that, we really have to figure out how animals are related. So we ended up doing a lot of molecular phylogenies. I did a lot of molecular phylogenies, both ascidians and hemichordates, and there is no fossil record really for either one of those, or very scanty fossil records, so we have to do modern technologies. And then continuing to look at deuterostome development and hemichordate development and evolution. So what are deuterostomes? You are here as a vertebrate. Tunicates, hemichordates, cephalochordates. I'm going to show you the evidence that I don't believe that Xenotrabella, or that what they call the Xenoacoelomorpha, are deuterostomes. We have echinoderms. And then the lophophorates have gotten kicked out. They're in their own group, the Lophotrochozoa. Chaetognaths got kicked out. They were put into Gdysozoa and then Lophotrochozoa, and now they're considered sort of a basal bilaterian. So now that we only have the few groups, how did we get interested in hemichordates? I was doing a postdoc looking at metamorphosis, and there's cells going everywhere and things opening up and all kinds of cells dying in ascidians. And I looked at these and I was like, this is not the ancestor. Like, no ancestor would be this way for the chordates. I'm like, I got to go look for hemichordates and see if they really have gill slits and whether I think those are the right things or not, because these tunicates, we need to have cell markers for them, and we need to try to figure out where everything's going. And it's very exciting. It's a time now where a lot of people in the tunicate community are doing that, so we're learning a lot about it. But in 1995, I was like, I got to go find some hemichordates and see whether they're the ones to work on or not. And so this is a Tychodera hemichordate here. This is a Sacoglossid hemichordate. I got so into hemichordates, I started doing their phylogeny. I was telling everybody about them, and people are just like, oh my God, I've never heard of these things, and please. So anyway, I'll show you a little bit about them. The colonial hemichordates are really important because this is what they kind of look like. They're moss-like. They're very hard to find unless you know where to find them. And the reason they were important is that's what people thought was the deuterostome ancestor. And I never thought that was right, but I was like, I got to go find these and find out what's going on with them. So these are the different theories of chordate origins. It turns out that Garstang's theory, sorry, this is like a trigger thing here. Garstang's theory, maybe I can use this. There we go. Sorry, I know that's bad for the people on the side, but so Garstang's theory, everybody came from a beautiful auricularia larvae. That everyone would love to do that. These larvae are gorgeous, echinoderm larvae and hemichordate larvae, and it's easy to take the echinoderm larvae and make a hemichordate larvae, but it's really hard to make that next step up to a tadpole larvae. Chordate larvae, and everything we know about gene networks and how they work suggests that this is not the way that things evolved, even though it was a great theory. And Thurston LaCauley really believed this, and when he gave it up a few years ago, then we said, okay, we can probably give that one up. Romer, this is what was in most of the textbooks here in the United States, starting with this colonial hemichordate here that somebody taken the stolons off because they didn't know what they were. But then you go from a colonial little tiny moss-like animal to these solitary sexual animals. Works pretty well with an ascidian because they're also hermaphrodites. Most colonial animals are hermaphrodites. And so these are hermaphrodites and these are hermaphrodites, but these other ones are solitary sexual animals. And so it just never made sense to me that they were doing that. When we did phylogenies, and we started with 18S in 2000, and we published that actually the reason that hemichordates shared gill slits with the chordates, and they were called hemichordates because people thought they were half chordates because they had these gill slits, is because the ancestor had that. And if echinoderms and hemichordates are sister groups, just to read the phylogeny, the ancestor had to be worm-like. And so we did the phylogenies, and we published this in 2000 in PNAS. And what I said there was further developmental studies will be important in revealing how the evolutionary successful chordate body plan may be evolved from a worm-like deuterostome ancestor. And I gotta tell you, that's when the crap hit the fan. Because people were like, what? I would give a talk at Friday Harbor. I was at Friday Harbor then, and somebody was in the back going like this. And I was like, I don't know what she's doing, but whatever. And then later my friend was like, you were so calm when she was back there saying this was all hand waving and crap. And I was like, oh my God, I didn't know that's what she was doing. I might have been mad about it. But talking to people, I'd say, what's your evidence that there's not a worm-like ancestor? Because we know that the Dicozoa and the Lophotrochozoa are both worm-like invertebrates. And people had been giving courses, starting with this colonial hemichordate. And Romer, unfortunately, I never got to meet him, but Bob Payne, who was at UW, he had known him very well. And he said, oh, he's a really big guy and had really great ideas. And so everybody believed this without really thinking that there could be a worm-like ancestor with gill slits. And then it's pretty easy to make a chordate from that. And so people continued to publish the other phylogenies. This is great, Carol, because I didn't realize I didn't have to answer questions. Now I can be a little tougher than I was. I was like calm putting this together, but now I'm like, oh, goody, that's great. So I think what happened was this. Oh dear. So what I think confused people is this whole coloniality. So in both tunicates and hemichordates, you have coloniality, and coloniality really confused a lot of people about these things. Okay, so this is just to show you the evidence that this group here, the Xenocoelomorpha, which by phylogenomics has been shown to be a deuterostome, and then there's other work. This is how beautiful they are, the Nemerdermatids, the aceals, and the Xenoturbella. You see how gorgeous they are, how they look a lot alike. They don't have nephrozoa, and so much of the phylogenomics shows them to actually be the sister group to the rest of the Bilaterians. They tend to be a little bit unstable, and phylogenetic programs do that. It's called long branch attraction. When there's things that are really different, what will happen is that our phylogenetic programs will put those things together. And anybody that tells you that their phylogenetic program is not sensitive to long branches, not telling you the truth, because all the phylogenetic programs are. So this is the paper out of Andy's lab here, Andy Heinel's lab. This is the what's now called the, I'll go to the next one here, I can see it better. The Xenocoelomorpha, they're the sister group to the rest of the Bilateria. So they do come out as deuterostomes sometimes. I think that's long branch attraction. So this is all with phylogenomics. And the problem with phylogenomics, I always thought if we had all the genomes, we could do this. Genes evolve at different rates. So you have fast rates, you have slow rates, you have kind of middling rates. And which ones do you use for phylogenomics? And I think we're still trying to figure that out. We still don't know the answer to that. Okay, so going back, there's other ways to look at these things, right? You can look at genes, you can look at groups of genes. We've heard a lot about the Hox genes. Echinoderm and hemichordates have a different posterior Hox. We have the 5, 9, 10, 11, 12, 13 that are found in all the chordates. In echinoderm hemichordates, they're called 9, 10, 11, 12, 13, 12, 13. And they're very different. These are not found in those Xenoturbella groups. is find these only in echinoderm and hemichordates. Tunicates are always the odd man out. They've lost a lot of their Hox genes. They're not expressed really very collinearly. And so it's complicated. So if you look at, this is HOX1 now, so this is another paper where they're looking at the Hox genes, and you look at HOX1, you can see that these xenocoelomorpha do have a conserved motifs that you don't find in the Amblycraria. So this is now Tony, my son that I went to Woods Hole with, and he was married, and he now has 2 daughters. And I'm going to go ahead and just kind of shut this down here. So we're going to talk about invertebrate gill slits with pharyngeal gill slits. And I'm just going to say that we've looked at all this developmentally. So, and Chris Lowe's lab has as well. So you see this anterior, posterior Hox specified body plan. They have a neural tube that rolls up. I'll show you that. We haven't found anything that's a notochord. And again, the brachyury has been around a long time in a lot of animals. And it turns out in the hemichordates, they have the 60 notochord genes that we found in the rest of the chordates. So we're still trying to figure out, is there some place we can find a network where those things are all together? But the pharyngeal slits that people noticed and called them hemichordates, clearly there. So when you put that on a phylogeny then, what you find then is these are the Pax19, which is in the gill slits that you see in all of the animals, except what we originally said was we think they were lost in the echinoderms. And not only are the gill slits there in hemichordates, but they're also, HOX1 is expressed right after the first gill slit. Which it is, of course, for all of you, your first gill slit's your job, but your HOX1 was expressed right after that first gill slit. And you also see hemichordates, not only do they use the same network of genes to make the gill slits, they make them the same place on the body plan, which suggests that they are homologous. And again, the echinoderms, kind of the odd man out, and the ascidians are kind of odd man out on this one. So if you look at these fossils of echinoderms, people had called these gill slits. I've looked at these fossils. They look to me like they had gill slits. I talked about that once at a meeting, and I had a echinoderm paleontologist apoplectic. But some people do think that these are gill slits. This was a recent paper where they were looking at all the different gill bars and also suggested that they've been lost in echinoderms. But what's really interesting is looking at these genomes in hemichordates. So you are here as a human. The hemichordates, and we also possess these gene clusters where all the gill slit genes are together in a cluster. And these are transcription factors here. And then you see that you also find those in hemichordates, and this is branchiostoma. And then surprisingly, this is a sea star here. It has the gill slits cluster in its genome, even though they don't have gill slits. And if you look at other genomes, this is a mollusk here, they don't have that same cluster. So we might be able to go into genomes and find things that used to be there and that animals have lost now, but we can see them there, which is very exciting. I'm going to skip through all this. Just to say, one of the things we're still looking at is nervous systems, and there's a nerve net that these animals have that most people haven't talked a lot about. And then this is the central nervous system in vertebrates and hemichordates. And I've spent my whole career, all 40 years of it, looking at central nervous system, dorsal vessel, endoderm. Where does this notochord come from in chordates? But if you look at this, and you know that losses is probably as easy as gain, maybe easier than gain, all you have to do is lose the notochord, and you can make something very much like a hemichordate. And then echinoderms went off in their old crazy way for their body plan. And so we think that's a real possibility now that we're looking at. So just to conclude, Deuterostome ancestor was a mud-dwelling worm with pharyngeal structures for filter feeding. All chordate features of Ontoperus hemichordates are adult features that develop after metamorphosis. Chordate ancestor was likely a direct developing worm that evolved a notochord and central nervous system. and a Scythian tadpole larva was a secondary evolutionary event. And then just to put that all on a phylogeny here, we have lots of losses and some gains. And then my granddaughter Allie just graduated from high school last Monday, and there's Ibby, and there's my son Tony, and so time goes on. And I just have one last mentor I really want to thank. This was Mary Rice. She took the embryology course in 1945, was director of the Smithsonian Marine Station, and she would say, nurture great staff, keep impeccable notebooks, and eat lunch every day. And before I did a sabbatical with Mary, I never ate lunch, and now I eat lunch every day. And this is my lab from 2014. Sorry, I know. This is Sean, who did a lot of the hemichordate stuff. Mike, who went on to do hemichordates. Xander, who did a lot of the Ascidian work. And I didn't get a slide put together like Dingy did where he had all the people on it. But I said, is there anybody here? And there's actually 2 people here that had been in my lab. One is Carrie Weber, who's at UCSD and gave a great talk, is a postdoc with Kim Cooper. And the other one is Lauren Lubbock, who's at Stanford. I don't see Lauren, but is working with Chris Lowe on hemichordates. And I'm very, very thankful to all the people who've been in my lab, and also to all the people I've met at SDB. I love Ida. I told her I was going to say that I did do a meeting. I was a local organizer at Vanderbilt in 1996 for SDB. That was the first year of the Conklin Award. And then I moved to Seattle in 1999, and Ida called me up and said, hey, we're having another meeting, 2001. Will you be an organizer again? And I was like, oh my God. So I've organized a meeting at least a couple of times. And I just want to thank you again for the award and for all the wonderful science. And I echo what everybody else says. We have to keep doing our wonderful science. We have to nurture our young people. We have to nurture all these great minds that we have. And we just have to keep going and do everything that we can to keep going. Thank you.